Hello everyone and welcome to my YouTube channel Perfect Lab. In today's video we will explore the disk diffusion method, which is a simple technique used in antimicrobial susceptibility testing. We will also see how antibiotic disks create zones of inhibition and how these zones help us to determine whether bacteria are susceptible, intermediate, or resistant to an antibiotic. So let's get started. So the disk diffusion method, or the Kirby-Bauer method, can be defined as a standardized laboratory technique which is used to determine the susceptibility of bacteria to antimicrobial agents. And this method basically measures the ability of an antibiotic to inhibit bacterial growth on an agar medium and helps clinicians select the most appropriate antibiotics for treating bacterial infections. So let's look at the principle of the disk diffusion method. So the first principle, or the first procedure or method under the principle, is to standardize our bacterial suspension by adjusting the bacterial suspension turbidity to that of the 0.5 McFarland standard. And once this is done, the standardized suspension is uniformly spread on a Muller-Hinton agar to produce a bacterial lawn and just allow to stand for some few minutes. And after allowing it to stand for these few minutes, we use paper discs containing a known concentration of antibiotics are placed on the agar surface and then incubated. So during the incubation, the antibiotics diffuse radially outside from the disc onto the agar as shown in the picture here. And as this happens, a concentration gradient is established around each disc over here, and as a result, susceptible bacteria are inhibited to produce a clear zone of inhibition as shown over here. So whilst resistant bacteria grow close to the disc as shown over here to produce a small or no inhibition growth. And once this is done, the diameter of the zone of inhibition is measured in milliliters using either a ruler or a caliper or a zone reader and recorded. So once we are done with the recording, we interpret our result by comparing our measured diameter of the zone of inhibition to or with the CLSI or the UCAS interpretive standard to classify the organisms as susceptible, intermediate, and resistant. So when the bacteria is susceptible, what it means is that the antibiotic is likely to inhibit the bacterium at the recommended therapeutic dose. And when it shows an intermediate result, so what it means is that the antibiotics may be effective under certain conditions, such as higher doses or when the drug concentrates at the site of infection. And when the bacterium shows a resistant result, what it means is that the bacterium is not inhibited by achievable concentrations of the antibiotic. And in this case, treatment usually is unlikely to be effective. So let's look at the materials involved or required to perform the disk diffusion method in antibiotic susceptibility testing. So the first is our pure bacterial culture that will be used to prepare the bacterial cell suspension, and it needs to be freshly grown between 18 to 24 hours. So the second is our Muller-Hinton agar plate The third is our sterile cotton swab. The fourth, sterile normal saline or nutrient broth. 0.5 McFarland standard. The antibiotic disc, a sterile forceps or disc dispenser, an incubator, a ruler, a caliper or a zone reader, and a marker for labeling plates. And one important information we must note is that all materials must be sterilized or clean, and we must use aseptic techniques throughout the procedure to ensure accurate and reliable results. So let's look at the procedure or the steps involved in the Kirby-Bauer methods or the disk diffusion method in the antibiotic susceptibility testing. So the first method or the first step is to standardize your bacterial suspension by adjusting the turbidity to that of a 0.5 McFarland standard. So once we are done with this, we inoculate the entire surface of the muller-Hinton agar plate using a sterile swab to create a uniform lawn culture. And in doing this, we swab in 3 directions by rotating the plate approximately 60 degrees each time. So we swab from this side of the muller-Hinton agar to this side. Once we are done, we rotate the muller-Hinton agar plate and swab from here to this side. And once we are done with this, we rotate it again and swab from here to this side. So once we are done with this, we allow the muller-Hinton agar plate to stand for 5 to 15 minutes. And this is to allow the bacterial suspension to be completely absorbed into the medium. So after doing this, we place our antibiotic-impregnated disc on the agar surface using a sterile forceps or a disc dispenser. And in doing this, we must ensure even spacing between the disc and firm contact with the agar surface as shown here. So we then incubate the plate at 35 to 37 degrees Celsius in an inverted way for 16 to 18 hours for most non-fastidious organisms and for 18 to 24 hours for most fastidious organisms. And what we must note here is that the incubation allows antibiotics to diffuse and inhibit bacterial growth. So after the incubation, we measure the diameter of the zones of inhibition in millimeters, either by using a ruler or a vernier caliper or a zone reader. So once we are done with this, we compare our result with the CLSI or the UCAS interpretive chart to classify our bacterium or organism as susceptible, intermediate, or resistant. So let's look at an example which is being provided by the CLSI for E. coli using the antibiotics amoxicillin. So assuming after performing our disk diffusion method, and it's like we measure the zone of inhibition for the antibiotics amoxicillin, and we had our value or our diameter to be equal to or greater than 17 millimeters. So what it means is that the E. coli is susceptible to the antibiotics amoxicillin over here. But if we are to measure our zone of inhibition, and it's like we have a diameter between 14 to 16 millimeters. So what it means is that our bacterium E. coli or the antibiotics, the amoxicillin, will be effective if higher doses is being used against the bacterium E. coli, and we normally record this result as intermediate. And if the zone of inhibition is equal to or less than 13 millimeters, then we record it as resistant, so which means that the bacterium E. coli is resistant to the antibiotics amoxicillin. So this is basically how we can use the CLSI or the UCAS interpretive chart to classify our organisms as being susceptible, intermediate, and resistant to the antibiotics that are being used in the Kirby-Bauer method or the Disk Diffusion Method. So let's look at the interpretation of result in the Disk Diffusion Method in the antibiotic susceptibility testing. So the result, susceptible, means the antibiotic used is likely to inhibit the bacterium at the recommended therapeutic dose. And the organism here usually produce a large zone of inhibition around the antibiotic disc. And the result, intermediate, means the antibiotic used may be effective if higher doses are used or if it concentrates at the site of infection. And the organisms here usually produce moderate zone of inhibition around the antibiotics. And the result, resistant, means the bacterium is not effectively inhibited by the antibiotic. So the antibiotic is unlikely to cure the infection when used. And the resistant organism here usually produce a small zone or no zone of inhibition around the antibiotic disc. Now let's look at the advantages and the limitations of the disk diffusion method. So let's start by looking at the advantages. 1. This method is simple and inexpensive. 2. It's easy to perform in routine microbiology laboratories. And 3. It's suitable for testing multiple antibiotics simultaneously. So for the limitation, this method provides qualitative results by classifying bacteria as susceptible, intermediate, or resistant rather than determining the exact minimum inhibitory concentration. And it's also not suitable for some fastidious or slow-growing organisms without modified media or conditions. And the result can be affected by the agar depth, inoculum density, incubation conditions, and the disk potency. So thanks for watching, and if you found this video helpful, please like, share, and subscribe to Perfect Lab. Leave your comments or questions below, and don't forget to scan the code on your screen to follow us on WhatsApp for more laboratory updates and tutorials. So see you in the next video.